Hello everyone. The topic of my talk is a viral hijack. HIV-1 exploits the host circRNA ciTRAN to overcome the transcriptional repression. We all are familiar with the central dogma of life, from DNA to RNA and then RNA to protein. That's how the information is transferred. But that is not the only function of RNA. Today we know a myriad of function that can be performed by this RNA molecule. It was until in 2012 where we found a new member of this RNA family called a circRNA formed by the back splicing. Now, back splicing involves the downstream exons binding to the upstream exons and forming a circular structure which can even include introns as well. There are multiple functions of this circular RNA which is performed under the different compartments of the cell. But this circumnavigated to the viral world as well, where this virus particle can form their own circular RNA and as well as induce some of the host circular RNA, which can be both proviral, can be antiviral. We are going to talk about the HIV-1 virus with 40 million people living with this disease at the end of 2024. A typical HIV structure looks like this, and where we have these three important structures: GAC, Pol, and envelope, inside which the core particles we have three more accessory proteins, which is VPR, NF, VPU, and also there are two molecules of RNA, which play an important role in the infection. So we sought to determine what are the circular RNAs inside the HIV-1 infection. Until 2023, there were no reports of circular RNA inside HIV-1 infection. So we utilize a nanopore direct RNA sequencing technology to form the circular RNA which are upregulated in the HIV-1 infection. And we found one of the circular RNA, ciTRAN, which is formed from the SMARCA5 gene, reduced the infection up to 50%. And we also found out HIV-1 VPR was able to induce this circular RNA. We utilized the mass spectrometer to analyze what are the proteins involved in sponging or decoying some of the proteins by the ciTRAN. We found SRSF1, a negative regulator of this HIV, which was responsible for the reducing this infection, which was further also confirmed in many patient samples. We then determined what is the mechanism behind how this HIV-1 VPR was able to induce this ciTRAN, and we found the TGF beta behind all the signaling cascade. This was further confirmed in the transmitted founder viruses as well. And in conclusion, what we found was packaged VPR was able to induce DNA damage, which downstream led to the TGF beta induction, and then the phosphoSMAD2/3 was able to induce this ciTRAN, which sequestered SRSF1, through which this HIV-1 promoter was able to transcribe effectively, and that's how the ciTRAN induction takes place and it reduces the infectivity. Thank you.